now that we have our list of differentially expressed genes, the next step is genome set enrichment analysis. So what you're doing here is you're looking in a dictionary to see what each of those 528 genes do. So they'll have different functions, they might belong to specific pathways, they will be found in certain cellular locations, so cellular compartments, and they'll be involved in molecular functions of biological processes. So what you're doing is creating a model where you randomly pick 528 genes and you're working out what's the probability of randomly picking 528 genes from each of these different defined groups of genes. Then you're comparing that to how the 528 are actually found. So what you'll find is that there might be some more in a specific pathway than you'd expect by random. So you can calculate a probability of this happening and by chance, and the actual probability of it happening, and you can work out the p-values. So you can do a hypothesis test to see if it fits a random model or if it's genuinely something interesting. So here we got the enrichment analysis. I place the cursor at the start of this and press run. So it's going to take, it says about a minute, but it might take a bit longer than that. And I'm going to click this screen back into plots rather than packages, because in a minute we're going to do some plots. So once it's done that analysis and split the data into different groups, you need to display them. Now I've got one here, a piece of code here which will display all of them together, but I'm going to display them each one at a time. So I go to the next one and I press run. So this is showing me the gene ontology, so that's the dictionary that it's using for biological processes. Now at the minute you can't read that, so let's do a zoom. So when I hit zoom it opens another window, in this case on a second screen that I have here, which I've just had to drag over. So it shows you that enzyme-linked receptor protein signaling pathway has 8 genes in it, and the probability of this happening by random is less than 10 to the minus 3. So here is the negative log to the power 10 of the false discovery rate, which is the equivalent to a p-value, but modified for multiple testing. So here it's 0.0001. So that is significant. The rest of these are a bit less significant. Glutathione biosynthesis, peptide biosynthesis. To be honest, there's nothing that stands out there as something you'd expect, and that's probably going to tell you an interesting story. But you've got that data. Next thing, we'll run the next one. So this one is cellular components. So this is going to show you where the particular genes that are switched on are acting. Now lots of them are acting in the extracellular region, 37. 21 of them are in extracellular region part, whatever that means, and there's some attached to flagella axoneme. Now that's interesting because flagella are associated with motion and movement of the cells. So that could be interesting because that could be associated with metastasis and the cancer spreading to remote locations. So there is the beginning of a story. Now if I run the next one, I've got pathways. I haven't got a lot here. I've only got 3 particular pathways. So I've got OCT4 in mammalian embryonic stem cells, LXR, RXR activation, and neuropathic pain signaling in dorsal horn neurons. This one doesn't make any sense, the neuropathic pain. I don't know what OCT4 is, and I probably need to look up what LXR and RXR are as well. So that is something that you can do. So if you go to Scholar and I go LXR, RXR and breast cancer, the pathway signaling is associated with triple negative breast cancer in African American women. So there is a known connection already, and that is a valid and important result. So you're getting significant results. Now, what I should also say is when you've got this particular plot open, you can also export it. So you can save it as an image, as a PNG, which by default it's called rplot. So this one you want to call, let's say, pathway plot, and it saves it as a PNG file. By default, you can change that to TIFF. So TIFFs are non-compressed, SVGs as vector graphics, and much better if you're going to do something like a poster. JPEGs are small, but they lose quality and resolution and can get pixelated. PNG the same. So PNG is the same as GIF, but without copyright issues for the technique. So I can save that. So now if I go into the directory, I've got pathwayplot.png, so I can open it this way. So this is something much easier to put into your documents. Okay, so that's done the second one, the third one, sorry, and here's the last one, which is molecular function. Again, I need to zoom this because at the minute you can't see anything. Oh, I have zoomed and it's not, must be showing me something behind here. So here it is. So I've got growth factor activity, ligand-gated ion channels, ligand-gated channels, heparin binding. Okay, that's interesting. So we know that heparin binding is associated with breast cancers. So again, I can export this plot, save as image. This one is molecular function. I can type the name molecular function, and I've got that plot, which if I open, ooh, it's a bit small. So, oops, I did the zoom though. That's an issue. So, what did I have? Have I got the zoom here? If I've got the zoom here, I can also do the save image in the zoomed one, which seems to be a better idea. So I wish to save that one, so it should be molecular function .png. So I'm going to overwrite the file that I've got, save, yes it exists already, get rid of it. Now when I look at it, it's readable. So you want to save it out of the zoomed image file, otherwise you'll end up with a very small image that you can't probably read. So those are the Go associations. But another way of visualizing your output is to look at the pathways themselves. So what I'm now going to do is go on to a more detailed pathway analysis.